So as I said, the big question today is to understand how do we solve a question if it contains one of these verbs. Let us start with describe. The verb describe. Describe. To describe something, it's something not easy. Describe يعني وصف. Can you really describe something for me? If you add إذا كنت عم بتزيد أنت وعم توصف بهارات معناتها you do not know how to describe. Because the describe is, as definition, it is to note all the details of the document. It is to note the details of the document. Only the details, what exists in the document without adding or eliminating any information. So, when I ask you to describe, you have to tell me exactly what exists in the document. You must not add information. You must not omit or delete or eliminate information. Exactly what exists in the document only. This is what we name describe or description. Let me give you an example. Suppose we have this document. Look, given the following document, and this document, what? Representing Mosier experiment. Mosier, it is a scientist. He is a scientist. This document represents an experiment, realized by Mr. Mosier. So, given the following document representing Mosier experiment, this is our document. And what is our question? Look, 1, describe this experiment. So describe, it is a verb. It is a verb we have to pay attention for. You have to pay attention when we have the verb describe. طيب, the question is to describe this experiment. Let us read together this document. What is in this document? What is this? Very good. It is a mouse. A rat, a mouse, whatever. It is a mouse. Look here. We have the spleen is removed. It means that we remove the spleen of a mouse. The spleen. The spleen is an organ, exists in the left upper side of our abdomen. Spleen. What is meaning of the spleen in Arabic? Do you know? The spleen is not the liver. منه الكبد لا ولا المصران لا. Spleen الطحال. Very good. الطحال. Spleen. طحال, we know. You know something. Sometimes خليني اقول لك sometimes after an accident بعد ما حدا يعمل حادث بيقولوا they removed his spleen. قاموا له الطحال تبعه. So mainly we heard by this انه حرام قاموا له الطحال. Okay, مش مشكلة. Is the spleen without any role in the body? No, it has a role. But لما يعمل الإنسان accident, spleen في بقلبه big blood vessel شريان دم كبير بيعمل هيموراجيا بيعمل عفوا نزيف داخلي. إذا كان ممزق السبلين وما انتبهنا ما أمنا السبلين وكويناه بيعمل نزيف داخلي وبيقتل. لذلك most of the times, مش sometimes, after an accident we have to remove the spleen to prevent the hemorrhagia and for saving the life of the individual. The spleen has a role in immunity, but it does not affect in severe way, its removal does not affect in severe way the immune system. Now what? As I said, we'll continue together in this document. We have this mouse and the spleen is removed. امناله اتحل هيدا الفار. Okay, we obtain, look to these two colors here. Cell suspension. Cell suspension means group of cells. How many types of cells do you see here? Very good. There are two types. Some are represented by the blue color, the others by the red or purple type. So, there are two types of cells. We say cell suspension, group of cells containing macrophages and lymphocytes. Yani some cells are called macrophages and the others are called lymphocytes. Now, look to these arrows. 1, 2, and 3. Here, in this container, we put the word macrophage. In this container, we have macrophages and lymphocytes. In the third one, lymphocytes. So, what we are doing by this with these cells? Yes, we are separating the cells in 3 containers. In the first container, we are putting macrophages. In the second container, we put macrophages and lymphocytes. And in the third container, we put only lymphocytes. So, I will repeat, in this experiment, in this document, we see that, we have a mouse. The spleen of this mouse is removed, and in the spleen we observe 2 types of cells. We obtain a group of cells, cell suspension, a group of cells containing macrophages and lymphocytes. Some cells are called macrophages and the others lymphocytes. We separate these cells in 3 containers. In the first container, we put macrophages. In the second container, we put macrophages and lymphocytes. And in the third container, we put lymphocytes. Now, what do we have here? Let us read them. Here we have this culture in the presence of SRBC. 4 days. Culture. In Arabic, culture يعني زراعة. بنقول عم نزرع الخلايا. استاذ شو معنات عم بزرع؟ يعني ببحش بالأرض بحط الخلايا وبزيد فوقهم مي؟ لا يا بابا. In order to culture cells, we put the cells with nutrition. So the cells will multiply. Only this. بنحط الخلايا مع أكل. بدي أعمل زرع خلايا، بحط الخلايا مع مكعب دجاج ماجي. وي مع مكعب دجاج ماجي. كانوا من قبل we put agarose or jello. بحط الخلايا على الجيلو، على طبقة من الجيلو. So the cells will absorb, will eat glucose and sugar from this jello and they will multiply. هيك بنسميه نحن عم نعمل culture. Nowadays we use ماجي. بيستعملوا مكعب ماجي لأنه في كتير nutrition بيخلي cells to eat and to multiply actively بسرعة أكتر. So what is important here? إنه culture يعني we add nutrition to these cells in the presence of sheep red blood cells. SRBC, it is the abbreviation of these 4 words. The first one, sheep. Second, red blood cells. Yani we put the red blood cells of sheep also. So, in this part of the document, we have culture in the presence of sheep red blood cells for 4 days. So we are adding nutrition in the presence of sheep red blood cells for 4 days. For each of these 3 containers. So if I ask you, what do we put or what did we put in this container, in the first one, it says, in the first container, we have macrophages. Only macrophages? It says, ايه macrophages؟ يا خيي بس macrophages تطلعهم بالدوكيمنت منيح. It says yes, macrophages and sheep red blood cells. And sheep red blood cells. So in the presence of sheep red blood cells, يعني here sheep blood cells are present. In the second container, sheep red blood cells also are present. We are adding them. And in the third also we added sheep red blood cells. So in the first container, we have macrophages and sheep red blood cells. In the second container, we have macrophages and lymphocytes and sheep red blood cells. In the third container, we have lymphocytes and sheep red blood cells. Good. So, this is the mouse. Its spleen was removed. We obtain a group of cells, cell suspension, group of cells containing two types, macrophages and lymphocytes. We put macrophages in the first container. In the second, we put macrophages and lymphocytes. And in the third, we put only lymphocytes. Then we add to them, to all of them, sheep red blood cells. We keep them for 4 days. And then, look to the last part of the document. What is the meaning of this arrow? This arrow, no antibodies. This arrow, anti-SRBC antibodies. This arrow, no. What is this? روعة روعة روعة. The result. Very good. يا عمي شو هالشباب والصبايا الجدعان. So it is the result. We do not obtain antibodies in the first container. We obtain anti-SRBC antibodies in the second container. And we do not obtain antibodies in the third container. Good. So, this is the first container, no antibodies. Anti-SRBC antibodies in the second container, and no antibodies in the last one. What did I did? Very good. I described this document. I told you what exists in the document without adding any information. طبعا ما في ضرورة أبدا ومش لازم حط role of the spleen أبدا. بس أنا كنت I was explaining for you what is the meaning of spleen. Not more than this. So I mean, if I want to answer, to solve this question, describe this experiment, I have to write this. Look to this. Now, okay, what? We remove the spleen of a mouse. Okay? تلاحظوا, look to this. We remove, or they removed, اللي بدكم إياه. We remove the spleen of a mouse. The cell suspension, look to this. Cell suspension obtained contains macrophages and lymphocytes. We obtain a cell suspension containing macrophages and lymphocytes. We are not adding information. Voila, we are not eliminating information. These cells, I will continue here. These cells, look to this. These cells are separated in 3 containers. I am writing what I saw in the document. Hide the mouse. The spleen is removed. We obtain a group of cells containing macrophages and lymphocytes, and then we separated the cells in 3 containers. These cells are separated in 3 containers. طيب. Then what? The first, okay, in the first one we put macrophages. In the second we put macrophages and lymphocytes. And in the third we put lymphocytes. In the 3 containers we add SRBC and we keep them in culture for how many days? For 4 days. So أنا قلت له للأستاذ, in the first we put macrophages, in the second we put macrophages and lymphocytes, and in the third we put macrophages. Then we added to the 3 containers SRBC and we put them in culture for 4 days. So I am putting the details. I am not adding information. I am not eliminating information. Then what? We observe that in the first container there are no antibodies. Look to this first, no antibodies. In the second, there are anti-SRBC antibodies. In the second, there are anti-SRBC antibodies. And in the third, there are no antibodies. In the third, there are no antibodies. So this is the description. It is not something that is hard. It is easy. المهم خاصة الصبايا يا شباب، خاصة الصبايا يا خيي بلسانهم بحب يتحرك، خيي لسانهم بحب يتحرك. بليز يا صبايا، بليز يا صبايا. When we ask you to describe ما تحطوا بهارات، ما تحطوا بهارات. Only write what exists in the document. Ça va? Y a-t-il des questions? Vous avez tous compris? Did you understand all? What is the meaning of or and how we solve the question or the verb describe once it is in the question? Inshallah.